All right, all right. I think we're about to start. Before I give the floor to your session leader, I don't want to take your time too long. I just want to promote two things which are important to us at Arctic Frontiers. So tomorrow, don't forget, there will be the Big Pictures science session at 1:30. So don't leave after today. Join us until tomorrow afternoon for the Big Picture science session. Plus, and I think there were already some questions about the virtual poster session. Don't forget, it's today, 5:30, and you can upload your poster, I think, until the minute before the session starts, and then discuss your poster and present it online. So that's all from me. Thanks, everyone, for coming and joining, and sorry to take all the time on important science discussion. So, Kit, it's all yours.
Well, Mats and I have the pleasure of welcoming you to session 5, part 2, on Atlantification. I think everyone in this room is very aware of the Arctic changing in very dramatic ways, 2 times the global average in terms of temperature, and in the Barents region, 4 times the average. So we're going to hear about implications and the physical changes that are ongoing in greater detail in today's session. I hope our speakers for today do justice to those yesterday, where we had a wonderful session together with speakers being extremely professional in their science, in their presentations, and in the timing of their presentations. But there is a clock in this room, so that will help us out even more. Without further ado, we'll call upon our first speaker, Dimitri, and he's going to talk to us about pan-Arctic modeling of gelatinous zooplankton.
So hello everyone, thank you for coming and thank you for the warm introduction. So I will present the topic of my PhD, pan-Arctic modeling of gelatinous zooplankton. The work was done within the group of Argel at the Alfred Wegener Institute under the supervision of Dr. Charles Harmans. So we many times heard that the Arctic is warming 4 times, 5 times different estimates stronger than the rest of the world. And so what happens also in ecosystems that the species are migrating northwards and their distributional patterns are changing. What is usually as a best indicator, because they're moving quite fast, there's zooplankton types of the organisms, and have like their different estimates moving with 100 kilometers per decade towards the north, some species. But the problem is that most of the studies, they're focused on the hard-bodied organisms like crustaceans, and usually the gelatinous zooplankton was neglected because there was a conceptual idea before that there's that end of the food webs. However, it was shown that recently, especially with new optical devices, that they're quite abundant and quite important for the food webs. So with this, the hypothesis is that the global warming will affect this gelatinous zooplankton communities, and our objectives are revealing the environmental drivers behind the structure of these communities, describe their geographical and vertical distribution, and also project the future changes of these communities. As a biological data, we selected 4 databases, OBIS, GBIF, JEDI, and Pangea. So for the Pangea, we had to dig a bit into the data sets, and we collected over 200 individual data sets, and they were mainly quite complementary, as you could see from this map. So they like different countries contributed to the different databases, and we end up with 10 most abundant species. As you could see, the most dominant group are hydrozoans, and especially Aglanta digitale, which is like really the most widespread jelly in Arctic Ocean. It's taking like a third of all observations. So the next group are Benicularians, Fritillaria borealis and Negapleura velhafei. Then there is a phylum of ctenophores, the Mertensia ovum and Piroia species, and the last ones, but not the least on impact, it's Pyrophilians and Negapilata. Those 2 might be quite dangerous for the fisheries in the Arctic region in the future, and actually right now as well. So our materials and methods, we pre-processed the data. That was quite messy. As you could see, out of 80,000 of records, we end up with 12.5 thousands. Then we took the FISM oceanographic model. This is from the SMIP6 assessment, historical runs, and also the future runs under the under SSP 370 scenarios meet strong emission scenarios. So then we had to unwrap this three-dimensional structure into 2.5-dimensional to fit to our model, and then we applied the Mach-Sand model. This is quite widely spread model, but it's doing really well with presence-only data that we had. And then we projected the current state, the action, and also the future one. On the scale of the environmental predictors, the most important was the sea ice, and explained mostly for like most of the species, the sea ice were explaining most of the variation in the data sets, and especially it was notable for peripheral and Antarctica. Yeah, but now we go to the special projections, and I will talk now about two species of hydrozoa. Those two, they're probably one of the most abundant in Arctic region, and two of cyphozoans because of their potential impact. So you could see on the graph, so this is a regular violent plots. You go from the depths 25 meters to 3,000 meters. So like you have 60 degrees, 90 degrees, so from the south to the north, and then you have on the left western and on the right eastern hemisphere. So what you could see that Cyanea capillata and Periphelia, they are both gaining the preferred niche conditions. So especially it is notable for the Periphelia, so it's almost like more than 2 times increase. And also what you could observe that's like this is reddish, the future scenarios, and the blue is the current state. So you see that in most of the regions they're not only migrating, but they're keeping their preferred niche conditions even where they persist right now. So now we will zoom into the most preferred depth layers, and this is 25 meters for the Cyanea and 460 for the Periphelia. And what we can see from the two-dimensional structures, that's for the Cyanea capillata, it's mainly like by the end of the century, it's occupying almost all the shore areas of the Arctic region, but Periphelia is mainly moving, so it's like historical, future. It's moving towards the Svalbards and the eastern part of the Greenland. The next class that I will discuss is hydrozoans. So those are actually from the same family, Rhopalmeae, and they have like their stratified like vertically. They're like having different niche preferences. And Aglantha digitale is type of more boreal species versus Antarctica is like truly Arctic, not truly, but more like Arctic species. And you could see that the Aglantha digitale becoming even more abundant through the time, and versus Antarctica is preferred niche conditions are shrinking by 30%. And again, we go into the two-dimensional structure, and what you could see is that basically Aglantha digitale was already found almost everywhere on mosaic expeditions, and so under the sea ice. But it will become even more abundant when we have like basically no ice in some months in Arctic. And Antarctica will shrink towards the central basin, so you could see in the future, and it will disappear in the Greenland part, in the Greenland Sea. So those are my conclusions. Periphelia is increasing together with Cyanea capillata, as well as Aglantha digitale, and Antarctica is retreating. So this potentially these changes will affect the ecosystem, food web dynamics. There are more food web models should be considered, but then we need to have also to know what they're feeding on, and those work is also is done within our group, and yeah, potentially these two could be really harmful for the fish stocks and the fisheries, and yeah, this data might be included in conservation management later. So, and thank you for your attention.
There is time for a question to Dimitri. There's a hand up at the back. Could you mic her?
Hello, and my name is Anna Karlsdóttir. I come from Nordregio and University of Iceland, and thank you very much for your very interesting presentation. I'm not a biologist, I'm actually human geographer. So I study regional dynamics, and I was in Uummannaq this summer and noticed that this species that is increasing was visible in the harbor and all around the Uummannaq fjord system. Can you simply explain what this means? What fish or mollusks or sea mammals feed on this species, the digitalis?
On
species?
Atlanta digitalia, yeah, we have actually the person in this room who is doing the like food webs, so who is feeding on whom. But Atlanta digitalis is yeah, consumed by the fish mainly. So like they are not like the dead end. So they are like consume copypods mainly, and I mean, yeah, that's more food web structures should be overlaid on the species distributions, because I can only say like from these models where they will go, but their impact, yeah, so it should be more like feeding should be added to these models. So, but yeah, but you're talking about Atlanta digitalia, right? So these small ones. Yeah, they're literally everywhere. They're like they, yeah. I mean, you mean their impacts? Yeah, so again, that should be more like food web.
So I guess that it will change the dynamics in the ecosystem in terms of fish species.
I think Atlanta will not really super affect this, but I mean, of course, they will compete with fish larvae in like this juvenile organisms for the copypods, of course. But I think like Cyanea and Spherophila, they can really wipe out all these copypods, so they have like really this huge tentacles. So I think those like bigger cyphozoans are the bigger threat for the fish rather than Atlanta digitalia, because fish could feed on the...
Thank you very much,
Sorry.
Dimitri. We will have 20 minutes at the end of session for more in-depth conversations. So I ask that our speakers remain in the room and spend that 20 minutes gabbing with folks with these extended interest questions. We must move on, and just a little warning to our speakers, the clock is counting down from 8. So when you hit 0, you have a minute or two to wind up. It's not counting down from 10. Our next speaker is Melissa, and she's going to talk to us about rapid ocean acidification in Arctic outflow waters in the last decade.
Thank you very much, and also thank you to my collaborators on this work. And most of them are in this room. And today I'm also grateful to present some of the changing chemistry that we observed in the outflow waters in the Western Fram Strait. And you have all heard about that the physics of the ocean is changing, but we see more and more evidence that the chemistry of the ocean is also changing. And the ocean has absorbed about 1/3 of the anthropogenic CO2, which has been a great favor for us. Otherwise, the atmospheric CO2 level would be much higher. But this is at the cost of the ocean's chemistry and has increased CO2, which has caused a shift in the carbonate system towards a less basic state. So we see decreased pH and aragonite saturation. Aragonite saturation is a dissolution capacity for aragonite, which is the most labile mineral that biological organisms form in the ocean. And so we follow aragonite saturation. I will mention it several times in this talk, since it's one of the indicators for progressing ocean acidification. The plot shows the global mean surface pH rates from many of more open ocean stations in the surface water. And also this is from the Hawaii time series, where we have longest time series both in the atmosphere and in the surface water. And we can see that most of this decrease you see here of almost 0.02 pH per year is following the atmospheric increase and the ocean uptake of this CO2. So we also see that ocean acidification is occurring in the Arctic, and there's more and more evidence, and you can see also this is from the Western Arctic, where this blue blue water consists of corrosive aragonite waters, and it's progressing further north and also shoaling. So you get a larger part of the water column which has these corrosive conditions. There's also evidence from more the Atlantic water influenced region in the Amundsen and Nansen basins, where this study here shows progressing it's hard to see the numbers, but it's per each 5 or 10 years, we see progressing anthropogenic CO2 signal in this water layer, which is getting more and more red. And in this middle part, we can see that the Arctic already has very low, in some areas, very low surface water aragonite saturation. So these shelves on Siberian shelves and the Western Arctic contains already high CO2, very low pH and low aragonite saturation. This water here is transported in the transpolar drift, crossing the Arctic basin and entering also the East Greenland current, and comes out in the western part of the Fram Strait. So we had some questions that we wanted to see. Can we actually detect this change in the Arctic? Can we see any of these low aragonite waters coming out in the Fram Strait? And can we see a change, and at what rates? And also, could we see this Siberian shelf water coming out? So fortunately, Norwegian Polar Institute already had lots of cruises or annual cruises going on to the Fram Strait between Svalbard and Greenland along 79 North. And we just added some of the carbonate chemistry parameters and focused on the upper water column, 400 meter of the water column, and looked at, we started in 2011 and we have checked on the data until 2019. So here is a section plot from the Svalbard towards Greenland. And so this is the Atlantic water inflow and the West Spitsbergen current here. And this is the outflow waters from the Arctic, where you can clearly see the more purple or bluish water here has low aragonite saturation and in the Arctic outflow water. So it seems like that we are capturing this Arctic outflow water in the western part of the Fram Strait. This shows then some of the results from 2011 to 2019, where we see changes. This shows the water column upper 400 meters towards year, and you can see increasing PCO2 throughout the water column, and also decreasing pH. And this pH in the surface water, we found the largest pH decrease rates, which were 3 to 4 times faster than the global mean pH decrease rate. This coincided also, we could see that the surface PCO2 was increasing by 5 microatmosphere per year, and you can compare that with the anthropogenic CO2 increase in the atmosphere of about 2. We also looked at the surface water aragonite saturation and could see that this decrease in aragonite saturation is also about double or 3 times the global mean. So this means that the atmospheric CO2 uptake of 2 cannot fully explain our decrease rates, because then it would be much lower. So there must be something else explaining these decreases that we see. And we know from other studies that freshwater acts in 2 different ways. It dilutes buffering ions in the water, particularly in the surface water, and that in itself enhances OA by just removing, in a way, carbonate ions. We have also seen, studies have shown that the Eurasian River discharge has increased, especially during the last decades. And also with fresher water, you increases the CO2 solubility. So in a way, you also get, with more open areas in the Arctic, with the sea ice loss, you also have waters that have a greater potential for ocean CO2 uptake. So we think that this is part of the story explaining why we see much higher rates than just because of the anthropogenic CO2 signal. So to sum up, yes, we see progressing acidification, and it's probably a signal from the Siberian shelves, we think, since most of the water is transported. I cannot get back. Okay, yeah. Oh, now you see me again. Okay, I can sum up anyway. No, so yes, and we can see, this from straight cruise is really, oh, great. Fantastic. Yeah. So, yeah, and we think that, of course, we have part of this anthropogenic CO2 is part of the story, but it's not only part, but we also think that freshwater dilution is diluting the buffering ions, and potentially also see increased meltwater-induced CO2 uptake, and that we also love to go to Fram Strait because it's so interesting area and very important time series that we have been building up for the ocean chemistry since 2011. So and it really captures this change within the Arctic Ocean. Thank you. No, it says 0.
Melissa has left time for questions if there are any. It was so very clear that they
Yeah,
are mesmerized.
Chemistry is, no one understands.
Our next talk is going to be presented by Samuel Lobet, Two Sides of Svalbard, Underwater Soundscape and Marine Mammal Communities Characterized Through Acoustics Data. And Samuel is presenting electronically for us today, so it depends on the back of the room hooking us up. Welcome, Samuel. We see you clearly.
Hello, everybody. Do you see my screen?
We do, but you're in presentation. You're not yet in full presentation mode.
Yeah. Okay. What about now?
Perfect.
Super. So, hello, everybody. My name is Samuel, and today I'm bringing a topic that I've been working on the last year at the Norwegian Polar Institute. I'm going to present two sides of Svalbard, underwater soundscapes and the marine mammal community characterized through acoustic data. The aim of the present study was to provide new insights about the marine habitat in west and east parts of the archipelago using sound data from passive acoustic monitoring, focusing our effort in three main fields of action. The first one was to characterize the underwater soundscape in the west and east parts of the Svalbard. And for this, we wanted to identify the components of the soundscape. I provide baseline soundscape metrics, so quantifying the amount of sound energy produced at the different time and frequency scales by different components. The second was to characterize the marine mammal community through acoustic data, so identifying species present seasonally and the timing of breeding when possible. And the third one was to quantify the marine traffic using AIS data as a preliminary step to see how well this spatial data links with sound detections by us. For this study, two locations with underwater recorders at opposite sides of the archipelago were chosen. The first one, Konsfjorden in western Svalbard, where a substantial Atlantification has occurred in the last 2 decades. From this location, an entire year from 2017 to 2018 was the latest available and the data chosen. And N2 in the eastern part of the archipelago, where more Arctic conditions remain. For this location, the data available was from November 2019 to September 2020. The methodology used in this study can be summarized in 4 fields of action. The first one would be acoustic detections. So vocalizations from all possible marine mammals were manually detected, also from vessels and air guns. So spectrograms of each file were visualized manually and scored. The second one would be soundscape analysis. So different computations were applied to all the data, including long-term spectrograms, power spectral density plots, and sound pressure level plots. The third one would be the marine traffic data. We use data from an autonomous identification system database to count the number of vessels around the recorders. And the last one would be the environmental variable, sea ice in this case. Sea ice data was included so we could link the sea ice data to the presence of endemic marine mammals and to the noise at different frequencies. A total of 11 species were acoustically identified, combining both locations. Each of the cells here represents the percentage of files with presence of the species in a given week. So at Consfjorden, walrus and bearded seals were frequently detected during the winter and spring, respectively. The seasonal visitors, in general baleen whales, were occasionally detected in late summer and through the autumn. And the vessels were detected constantly throughout the year, except during the winter. And the detections reached a maximum rate during the summer. At M2, the Arctic residents, walrus, bearded seals, narwhals, and bowhead whales, were detected throughout most of the year. Other seasonal visitors were sporadically detected, but we need to highlight the presence of fin whales for almost 2 months during the summer. And the vessels were only seldomly detected, and air guns were picked up for 3 weeks in a row during the summer. Here I present the results of the soundscape analysis, beginning with the long-term spectrograms, which is a representation of the sound intensity in color across a frequency and time axis. This allows to identify main processes producing sound in a long-term scale. So to help visualize the processes, I have highlighted them with ellipses divided in 3 main categories: geophonies in black, biophonies in green, and anthropophonies in red. In Consfjorden, we need to highlight the effect of geophonies on the sound energy, with glacier melting and runoff producing a high-frequency signature present for most of the year. Vessels can be detected as a bracketed low-frequency signal at different times of the year, although most notoriously in a long-term scale during December. And no biophonies were detected in a long-term scale at Consfjorden. At M2, we can observe a high-intensity sound detected during winter caused by the drifting ice. The loudest period of the year is related to the sea ice in April, indeed. Biophonies are clearly detectable and increase the sound levels throughout the year. Bowhead whales, bearded seals, and fin whales caused a signal that can be detected in a long-term scale. Human noise was reduced. We can pick up air guns in a long-term scale, and even one passage of a vessel in late summer. The power spectral density plots are very useful to understand the probability distribution of the sound energy across the frequency scale. So in this case, the plots include the whole year of data for each location. At Consfjorden, we can observe the maximum median sound energy at high frequencies with a bell shape. This is very characteristic of glacier ice fjords. We can also observe a multi-peak pattern with the vessel noise, with harmonic tones, which are very common around 100 Hertz in the case of vessels. At M2, the sound energy was centered at low frequencies due to the persistent ice noise for most of the year. But we can also observe a bulge in the 200 and 600 Hertz. And this is due to the bowhead whales and bearded seals localizations constantly. We can then confirm that biophonies have a relevant impact in the eastern Svalbard soundscape. We use sound pressure levels to link sound energy at defined frequency bands with different sound sources, in this case, the sea ice. From the results of this study, we observed that sea ice is a very significant factor in sound production in the Norwegian Arctic. At Consfjorden, we observe a drop in the sound pressure levels when the ice cover is at its maximum, and this is due to the sea ice acting as a shield from other noise sources, like it could be the wind. The frequency bands centered at 63 and 125 Hertz in blue and green are considered very good proxies of vessel noise. And we can observe an increase during December when the vessel acoustic detections were constant, as I showed you in the previous slide. A general increase of values in all bands occurs through the summer, which is generally a noisier period, and also when the traffic is at its maximum. At M2, we should highlight that when the pack ice break, it causes a general increase in the sound pressure levels. The last part of the results is the marine traffic that we studied using AIS data. Here we can observe some of the main differences between locations, with Consfjorden showing almost no traffic in winter, but with more than 20 boats within 50 kilometers in some days during the summer. And the trend of acoustic detections in general match very well the number of boats, except for December, when few boats cause this almost constant acoustic presence for a good deal of the month. At M2, the traffic was very low. We need to keep in mind the differences between scales among plots, but for most of the year, there were less than 4 boats within 100 kilometers. And to sum up, there was a great diversity of marine mammals acoustically present in Kongsfjorden. At M2, there was a dominance of acoustic presence of Arctic endemic species. The soundscape at Kongsfjorden was mainly shaped by glacier melt, whereas the main factor generating the soundscape at M2 was the sea ice. At Kongsfjorden, there was little impact of marine mammal sounds on the soundscape, whereas there was a high impact of marine mammal sounds on the soundscape at M2 in the east. And then a significant level of marine traffic had an influence on the soundscape in Kongsfjorden, although it was moderate. And at M2, the marine traffic had a very small presence. A very important finding was, although not the main focus of this presentation, that M2 constitutes a potential breeding ground for Spitsbergen bowhead whales, and a prolonged presence by narwhals was found at this location. So thank you to all parts involved in the study, and thank you for listening.
And thank you, Samuel, for taking the time at Madrid to present for us here today. There is time for one question.
Hi, thank you for this really interesting talk. I'm not sure if you're too much focused on sea ice, but I was just wondering if the frequency of your soundscape could be used to look at other sea ice variables such as concentration, and could we correlate, do you think if we could correlate your sea ice results with other results and confirm other observations on sea ice? Thank you.
Yeah, absolutely. I think there's quite a bit of scientists doing this in other parts, including in Svalbard, in Hornsund. There are some studies on glacial calving events, also in Kongsfjorden by the Indian team. So it is completely possible. And if now we include machine learning techniques, I think we will have a huge advance in this technique. So we could very possibly link sounds to the sea ice, different variables. Yeah.
Thank you very much. We'll move to our next speaker. And Carice is going to talk to us about deep sea larvae in a changing Arctic.
Hi there, everyone. Thank you for having me. I'm Karis. I'm from Woods Hole Oceanographic Institution, where I'm a PhD candidate working on early life history ecology of benthic invertebrates. And benthic invertebrates have complex life cycles. Many of them have a adult living on the sea floor, and they produce a larvae that goes into the water column for some period of time before coming and settling and recruiting to the sea floor. And I've seen a lot of great talks this week that are very big picture synthesis of long-term data, finally pulling together a lot of this Arctic research into very actionable policy recommendations and things like that. This is not that work. This is absolutely the beginnings of looking at deep sea larvae. This is the first time we've even attempted it up here. So it's really some baseline information for y'all. I have some pictures and not much else. But essentially, we are looking at the deep Fram Strait. So what's going on down there? The Germans at Avi especially have done a great job mapping the seafloor for benthic invertebrates in the Hausgarten LTER over the last few decades. One of the things that they've found, there was a great 18-year study from 99 till 2017 that saw that deep sea recruitment was really low. And recruitment is that transition from the larvae to the seafloor and actually growing up enough for the researchers to see it. They only found 13 species over an 18-year period compared to the 65-ish morphotypes that are seen on close-by reefs. So really low recruitment. Meanwhile, recruits also, the ones that did come, didn't even match the adult populations that they were seeing there, with only 3 of the most common species being present on the recruitment panels. And then finally, to understand recruitment, we also have to understand the larval populations, and very little is known. a lot of people say little is known. I think this is actually nothing is known of the larvae in that area. There's been some great work on larval abundances in the Arctic and distribution in other areas. We heard a great talk from Raphael yesterday from the Barents Sea. There's a great study from the Chukchi Sea. But these are studies looking at the larvae up in the water column and over the shelves. We are specifically looking at the larvae at the sea floor. So we essentially went out to say, who is down there? Where are they? What are they doing? We aimed to do 8 different sites in the house garden. This is Svalbard on the right side in the middle of the Fram Strait. Some technical issues and equipment loss to the seafloor. We only got 4. But for each of these sites, between 1,500 and 2,500 meters of depth, we deployed a lander. So this lander goes down to the seafloor. We detach from it, and it sits there for 24 hours with this pump. I didn't have a picture, so I drew it. And it pumps water, in this case, about 25 to 30,000 liters of water over the 24-hour period. And again, this hadn't been done, so we really had no idea what to expect. So it comes back 24 hours later, and we get basically a mesh. We use 64 micron mesh. Overall, we found about 30 to 50 meroplankton individuals in each of our 4 samples, with about 10 to 15 morphotypes in each sample. So we sorted them live on board and sorted them into ethanol, which we took them home to do genetic analysis with the CO1 locus to identify them to species. This is, again, very baseline stuff. It's a work in progress, but I have a couple little stories for you. So first was we found a ton of these embryos. They were called peach embryo. My advisor called them orange embryo. And sometimes we called them pink embryo. They were all the same thing. And they were all Borealisation maxima, which is an ostracod, which we were not as excited about. Some of you plankton people might be excited about ostracods. I think they're one of my favorites, but they are not a macrobenthic species. But the year-round reproduction was suspected for them, so here's a little piece of the puzzle. They are reproducing in June. Now we know. But overall, there was interesting biodiversity, and if we remove the embryos from the data set, there were still quite a few. We had maybe 5 to 10 morphotypes of actual larvae in each sample, and there was maybe 5 to 15 individuals per sample. But a few of the ones that we were able to get a better idea of who they were, there was a nemartine, or Stydia, which has been identified in the Norwegian waters, but not necessarily in the Fram. So not sure if this is just a lack of knowledge on the adults there, or whether or not this is a species moving into the space. We have what we thought was a Pelagosphaera. It actually matched Metridium senile most closely. Metridium senile is also not in the Fram Strait. It's a sea anemone, but the most common sea anemones, Ampharanthus and Gersemnia fructuosa, in the Fram are not in the genetic databases. So this is an example where we find something that has a really good read online, but we can't actually pair it to the species that are present because a lot of those adults haven't been entered in genetically. This guy I didn't actually identify yet. It's a bivalve, but we found it at all 4 sites in the deep sea, and then also in all of our surface tows. So this is either a case of a deep sea organism that's sending its larvae to the surface, or a shallow water organism that's losing larvae to the deep sea. And so by sampling at both depth and at the surface, we can start to sort of parse that out, but the bivalves are giving me trouble. And then the polychaete we found was a polynoid. It matched most closely to a South Pacific polychaete, which I do not think it is. But there is a polynoid by Glides groenlandicus in the Fram, and it is also not on the database. So honestly, much of what I was learning here is that a lot of the adult organisms that we know exist in the Fram don't necessarily have good genetic coverage online for us to be able to compare these larvae that we can't really understand morphologically in the way that we can with adults. So not only do we need to collect more larvae to see what they look like and where they are, but if anybody's collecting adults in the Fram, please preserve them in ethanol instead of formalin so we can identify a few of them with genetics. And then finally back to the all those embryos, another slight disappointment for me, a benthic ecologist. It was Metridia longa, which is a copepod. I personally don't like copepods. They get in the way of all of the meroplankton, so that was a little depressing. But yeah, so overall, we found tons of embryos. So there's a lot of things going on right near the seafloor. There is a lot of reproduction happening. This was in early summer. There were a few infaunal organisms showing up in the larvae, so the nemertines, the polychaetes, and the bivalves. So there is reproduction going on, but a lot of what we were finding was benthic-associated fauna. So maybe the benthic organisms are sending their larvae further up into the water column to feed. Maybe this was the wrong time of year, not really sure. So overall, we found almost nothing of the epibenthic megafaunal community that we were looking for. So that's what's been so heavily studied. That's what we know the distributions of. That's what we know wasn't really recruiting. And so at first this was really disheartening, but honestly it lines up with the fact that we weren't seeing recruitment in the area. So originally we were thinking maybe things are settling and experiencing post-settlement mortality, and that's why we're not seeing the recruitment. But maybe there's just not very many larvae in the water column. Maybe there's really low reproductive success at that stage, and the larvae themselves could be one of our bottlenecks. Overall, I think this raises more questions than answers, and we still really don't know much about how this is working in the deep ocean of the Arctic. But understanding this full life cycle gets us a better idea of how these populations might respond in the future. If we don't know how they're reproducing and how they're repopulating their own distributions, then we can't really predict anything. So I think this is a really important phase, and again, it's just getting started. And then finally, before I finish up, if anybody here has gone through plankton nets and has any idea what this is, please come talk to me. It's causing me problems. Yes. And with that, I would love to thank all the people that helped out with PS 126 on Polarstern last summer. Thank you.
I didn't recognize that there was such speciesism in the larval community. But time for one question. Can Colin have the mic, please?
So what about timing of year? So what would you expect if you weren't there in June, but maybe a little bit earlier?
Yeah, so I would love to go earlier in the year. Although there is, I think, a lot less seasonality of reproduction in some of these habitats that are less influenced by like the spring bloom and everything, although obviously they are getting the snow down to the deep sea. But I would love to do this on a broader scale. We have deployed larval traps to try to collect larvae year-round, so we won't get at when they were in the water column, but we can get whether or not they're there. Unfortunately, with the traps, just the way they work, we mostly end up getting all hard-shelled organisms, so the bivalves and gastropods, because they're what fall into the trap. But yeah, if I could get out on a big ship with a fancy lander earlier in the year, that would be wonderful. I would expect to see a little bit more.
Thanks very kindly.
Yeah, thank you.
And on to our next speaker. Sonia is going to talk to us about marine snow at the polar front, a feature of Atlantification.
Thank you very much. And contrary to the previous speaker, I actually do love copperpods, but I'm gonna talk about marine snow today. And this should be a nice picture of snow in Tromsø. It comes out a bit different here on the screen than it was planned, but the main point is that it looks very different than marine snow. And this study is done together with my collaborators from University in Tromsø and from Aquaplan, and in Poland from IOPAS. So this is how marine snow looks like, or some of it. So it ranges from 1 millimeter approximately to 10 millimeter at max. So we see this black bar should be 2 millimeters. So you can see that there are some large aggregates and some smaller, which look more like fecal pellets, like these ones, and then these larger aggregates. And all of those particles have an important contribution to biogeochemical cycles, and they are related to or influenced by how the water column is stratified. And all of these are influenced by Atlantification, of which we heard a lot about in the last session and yesterday. So I'm not going to spend much time on explaining the effects of Atlantification, but just so that you know that these particles do have an important role when it comes to Atlantification. And we studied those particles at the polar front, which you can see here in the I want to go back. Laser. Here in the map, this red line, I don't know if you can see it, but this is our study area in the Barents Sea. And you see two transects. On top we have salinity and below we have temperature. And basically on top the red is Atlantic water, and which is on the south of the polar front. And then we go to Arctic waters in the north, and there we also have a layer of very low saline meltwater. So the polar front itself is structured in a diagonal shape, with fresher waters on the north and more saline waters and warmer waters in the south. And there we also see stratification in the water column. We went there as part of a larger polar front study where we look at the entire ecosystem, and one of our objectives is to characterize the plankton community, including copepods, but also the size structure and spatial patterns at the front. And we went there during a time where we were expecting spring bloom. So that's in our areas between 75 and 80 north approximately. So the spring bloom should happen in May, June. And we were expecting a quite efficient transfer of lipids from the phytoplankton to higher trophic levels, including copepods. But what we did find was a lot of marine snow, hence the topic of this talk. And that shouldn't have come as a very large surprise, because already back in 2011, so 10 years ago approximately, we saw a lot of particles that we were thinking are most likely marine snow. So on the top here we see phytoplankton, so that's expressed as a chlorophyll biomass. And we have surface maximum with low values in the Atlantic water, and then a subsurface maxima at the Arctic side. And in the Atlantic part, in the left, on the bottom left, we saw a very high number of particles, which were measured by the laser optical plankton counter, which is a very good instrument for measuring particles in the water column. And for larger particles, we can also distinguish a lot of particles, including zooplankton and marine snow. But for smaller particles, we don't have that option so much. But based on some studies that have shown that we can use properties of these larger particles to know a little bit more about the smaller particles, we were thinking that these are most likely marine snow. So when we went back now in the last May and the year before, we had the sea glider that was equipped with an underwater video profiler, where we get much nicer images of the plankton and of the particles. So those images of the marine snow that I showed in the beginning, they were collected by this underwater video profiler. And we set it out in this area at the polar front. So this is a part of the Barents Sea. Here's open, and the glider was put out in the Atlantic part and in the Arctic part. So then we get a lot of images, really many. So we have 150,000, I think, from May last year or the year before, and then 600,000 from 2022. And what was apparent in last year is that there were like very, very many marine snow particles and very few copepods. So I don't know if you can read the numbers, but in those 130,000 images, there were 80 copepods or something. So it's a very strong contrast to what we see in the Norwegian Sea, for example, where we have very high biomasses of copepods. And then we had 80,000 marine snow or something like that. So in percent, it was 0.06% is copepods and 70% detritus. So these marine snow, as I said, it forms often during the spring bloom, and it can be aggregates from phytoplankton, but can also be fecal pellets and such. But these very fluffy marine snow and the different types that we could distinguish of the marine snow, they point to that there is a mismatch of the pelagic coupling at the polar front. So in all of those years, in 2011 and in 2021 and 23, it seems that marine snow is really, or large fraction of marine snow is a part of the Atlantification or of the Atlantic part, and that can, with an increased Atlantification, we therefore think it's likely that that may spread further as well. This figure shows different types of particles that we have characterized, and what you can also see is that there is some change in particles with darker, smaller particles in the deeper layer. So depth is on the left side, on the x-axis here, on the y-axis, and then some other brighter particles in the top layer. So there is some retention in the upper layer as well, and some transformation of the particles. So that's things we are going to look at in the future more closely, and also in relation to these biogeochemical cycles. So, but the mismatch that we are thinking is due to that there are a limited number of grazers at the polar front during the time of the spring bloom, because many of the grazers, which are copepods, are coming in with the Atlantic water from the Norwegian Sea, where they are overwintering. And here, in the study from 20 years ago, we see that there is a very high degree of advection, for example, in June, early June, late June, which then has not yet reached the polar front during the time of the spring bloom. So during the polar front study, we also deployed a sail buoy that was set out during the end of the cruise and stayed there for a month or 6 weeks after, and was equipped with acoustic high-frequency broadband sensors. So based on those data, which we have analyzed a little bit so far, we see indications that the copepods do come into the polar front a few weeks after we were there. But then we have a mismatch situation. So the consequences are, of course, that there is more material available for benthic organisms. And here you see one images of the UVP of some benthic creature or part of a benthic creature when the sea glider was very close to the bottom. So I do not know what a creature that is. If anyone has an idea, please let me know. But the particles in total, of course, also have consequences for cycles and for Atlantification. So with that, thank you very much for your attention.
Thanks very much, Sonia. I think we have to move along to stay on program here.
Okay.
With our next speaker, Elizabeth Jones, coming up to speak to us about inorganic carbon and nutrient dynamics in the marginal ice zone of the Barents Sea, implications for ocean acidification.
Great, thank you very much. I'm very happy to be here today to talk to you about some of the work we've been doing. And I'd like to acknowledge the effort of the co-authors and many others that have been involved in this work and the cruises as part of the Nansen Legacy project. So we've heard a lot about Atlantification and the importance of the Atlantic water inflow into the Barents Sea as a main transport mechanism for heat, salt, carbon and nutrients. It has strong influences on the sea ice cover, upper ocean physics, biogeochemical cycling, and the marine ecosystem. But we wanted to ask the questions of, well, what are the dynamics of the carbon and nutrient cycling in this region and on different timescales? What will be effects of ongoing warming, sea ice losses, and Atlantification? And what are the implications of these changes on ocean acidification of the Barents Sea? So through the Nansen Legacy, we occupied the transect in the northwestern Barents Sea with a series of 7 process or P stations. These extended from the Atlantic water domain in the south, which is south of the polar front, across the shelf of the Barents Sea and the Arctic domain, and across the slope and into the deep Nansen Basin of the Arctic Ocean. And through this, we were able to look at seasonal and interannual variability and the key driving processes of carbon and nutrient cycling. So firstly, a little bit of carbon chemistry. So CO2 in the atmosphere dissolves in the surface ocean. It forms carbonic acid. Now this carbonic acid dissociates or separates to form a bicarbonate ion and releases hydrogen ion. The bicarbonate dissociates again and forms a carbonate ion and a hydrogen ion. The 2 of the variables that we can measure in seawater is the dissolved inorganic carbon, and this is the sum of the main inorganic carbon species in seawater. Increases in the content of dissolved inorganic carbon increase the risk of ocean acidification. One of the other variables to measure is total alkalinity, and this is an indicator of the buffer capacity of seawater to counteract acidity. Increases in alkalinity help to decrease the risk of acidification of seawater. So firstly, I'd like to look at some seasonal variability, and we had 2 cruises in order to do this. So I'm focusing on one cruise we had in summer in 2019 and one during the winter-spring period in 2021. So the figures on the right show the sea ice cover, and in summer we had the ice cover in the northwestern part of the section, and in winter-spring the ice had extended across this part of the Barents Sea with the ice edge moving south. The plots on the left show the concentration of nitrate and dissolved inorganic carbon in the upper 250 meters of the water column along this section in the Barents Sea. We can see that during the summer there was very low concentrations of dissolved inorganic carbon and nitrate, tightly coupled to the upper surface layer that had been impacted by sea ice meltwater inputs, but also biological carbon and nitrate uptake from the seasonal primary production. The plots on the right show the relationships between inorganic carbon and nitrate, and silicate and nitrate, with respect to salinity. And this gives us a nice indication of the cycling of the carbon and nutrients during the summer. We can see that there were very high carbon to nitrate ratios, and this is indicative of the uptake during the primary production during the growing season. There were very low silicate to nitrate ratios, and this is a result of the progression of the blooms in the region, so primary production by diatoms, which will remove the silicate from the water column. Now we have a look at what's happening during the winter-spring period, and again, the plots on the left showing the nitrate and inorganic carbon distributions across the section, and the cycling of carbon and nitrate and silicate and nitrate in the plots on the right. So you can see the water column is much more homogeneous. There had been winter mixing that had enriched the concentrations of the carbon and nutrients, as particularly in the Atlantic water-influenced areas in the southern and northern parts of the section. This is a result of the remineralization of the organic matter and also greater Atlantic water influence in this well-mixed water column. As a result, the salinity shows much less variability, and we had low inorganic carbon to nitrate ratios, and this is a result of the remineralization of organic matter, returning these the concentrations back kind of to pre-bloom conditions. High silicate and nitrate ratios also are indicative of the fact that the diatom frustules dissolution returns the inorganic silicate to the water column and continued mixing. So we can have a look at what these changes are being driven by, some key processes, and on a seasonal perspective. So the plots here show inorganic carbon changes in the surface part of the ocean, So the surface layer relative to subsurface waters. Summer on the top and winter spring on the bottom along the section. So we can see that the summer is dominated by huge decreases in inorganic carbon, largely due to biological processes, so biological uptake of the inorganic carbon during primary production, particularly in the ice-free waters. In and around the summer sea ice cover, we see that there are large decreases in organic carbon due to the input of the ice meltwater, which acts to dilute the ions. In winter spring, you can see that there's much less change in the upper ocean, as the water column is much more homogenized through mixing. The changes that we do see in the inorganic carbon actually result from inputs from other processes, for example, rejection of CO2-rich brines from the sea ice. Now we have a look at alkalinity, and you can see again huge changes during the summer and smaller changes during the winter-spring period. And these were largely driven by input of sea ice meltwater during the summer. So we see that there's strong seasonality in inorganic carbon and nutrient cycling by key drivers of biological processes, meltwater inputs, and mixing. But what about the interannual variability? So we had a chance to explore this by looking at 2 cruises carried out at the same time of year in consecutive years. In the first year, in 2018 summer, we had much less ice cover relative to the summer 2019, where the ice edge was still the heavier ice cover persisted in the northwest. So again, I'm showing the changes in the upper water column, firstly for inorganic carbon, now between these 2 different years. So you can see in summer 2018, where we had much less sea ice cover during the summer, there had been greater amounts of biological carbon uptake due to the relatively longer growing season and relatively later stage bloom conditions. Now we have a look at the changes in alkalinity, so the buffering capacity, and we see that the scenario in 2018, where there had been less sea ice or earlier sea ice melt, presented much smaller changes in alkalinity, and these decreases are due to less sea ice meltwater input at that time. So this allowed us to start to look at what might be representative of an Atlantic regime, greater Atlantic water influence that resupplies the surface layer with inorganic nutrients to help fuel biological carbon uptake, with inputs of alkalinity that act to buffer against acidification. In a more Arctic regime, with more Arctic fresh conditions, we had more early stage blooms and less carbon uptake, where sea ice meltwater and fresher Arctic water presence reduce the alkalinity and enhance the risk of acidification. So we then went on to just speculate that maybe future warming, losses of sea ice, and this process of Atlantification in the Barents Sea could increase the biological and geochemical drivers to counteract ocean acidification in this region. Thank you for your attention.
There is time for a question.
Can you hear me? Oh, okay.
Yes.
I was wondering, do you have an explanation for the lower nutrient inventory in the central Barents Sea after the winter? So in the March cruise, you see that the nitrate was, well, 2 to 4 micromoles lower than in the Atlantic water on either southern and northern side. So do you have some explanation what sets the upper limit within the central Barents Sea? Is this reduced mixing with the deeper Atlantic water because you have a higher nutrient content of Atlantic water in the central Barents Sea at depth?
That's a very good question. Yes, you're right that we see, I think that it's that central part of the Barents Sea just exhibits very low seasonality within the nutrients and the carbon. It's predominantly Arctic conditions in all seasons, and they're generally characterized by lower nutrient concentrations relative to the Atlantic water influence stations. So particularly the inflow parts in the south and in the north are just being resupplied with much more Atlantic water-derived nutrients compared to the really Arctic-like conditions in the central part. but yeah, we're looking into more details as we're working on the writing up the papers on this.
Thank you very much.
Thank you.
And our last speaker for this session, Heather Canopy, and she's going to talk to us about biophysical interactions along Atlantic water convective pathways in the northern Barents Sea.
Okay, thank you. Yes, I'm going to talk about our attempts to use bottom-mounted echo sounders to investigate temporal variability in pelagic macroplankton and fish over timescales which can't be achieved using a ship. So 3 bottom-mounted echo sounders have been deployed in the northern Barents Sea at the locations indicated by the red dots. And I will focus in this talk on the A2800 mooring on the northern continental slope, where it's influenced by the Atlantic water boundary current, and also on the M1 mooring located in the Kvitøya Trough, where it's influenced by Atlantic water inflows from the north. And the instrument we use is the Nortec Signature 100, which is an integrated multi-frequency echo sounder and ADCP. And all of the moorings are located near other moorings which are collecting a range of physical and chemical data. So once we have the data from the echograms, then we have information on the density distribution of organisms, but by comparing the relative frequency responses, then we can also begin to learn something about the types of macroplankton that we are observing, as well as fish. Oops. So I'll begin by talking about the A-Twain mooring on the North Shelf. Here you see ADCP data. So this mooring is located within the Atlantic water boundary current, which is a very persistent feature throughout the water column. But what I want to highlight here is that this current also exhibits a lot of variability seasonally and at shorter term timescales, and this influences the water column structure and also the type of pelagic organisms that we find. So here you see data from the A-Twain mooring, temperature and salinity in the top plots, and below that the echogram recorded at this station. This is between December 2019 and the autumn of 2021. And I've shown for comparison the sea ice and day length below that. And looking at the echogram, the key feature that jumps out is the existence of this very persistent, thick mesopelagic sound scattering layer, which is typically more than 200 meters thick. It contains a mixture of mainly macroplankton, but also fish. And it varies in terms of its center of mass, thickness, and density according to the water column structure. So this is located primarily within the core of the Atlantic water boundary current. And please note the different vertical scales in the temperature and salinity plots and the echogram. So although the positioning of this scattering layer is related to the water column structure, and I was going to point out, interestingly, when the shelf edge current almost disappears and the warm intermediate layer also disappears, then we see this layer rising up into the pelagic zone and staying there for a few weeks. But looking at the echograms on this scale, a lot of the detail is lost. So here you see some 5-day snapshots during different seasons. And now you can see in the upper 80 meters or so, which is within the cold polar water layer, there are a lot of fish schools, and these are a common feature under the ice throughout the whole period of our observations. And the fish are there. The fish are more prevalent when the along slope current is weaker. And in figure B, you can just see vertical migrations, which bring the deep scattering layer into contact with the fish. And then when the ice disappears, there's a rapid increase in the density of organisms present, and the sound scattering layer is more than 400 meters thick at this time and extends up into the upper 50 meters. So we've made what is a preliminary attempt to identify how the abundance of some different macroplankton groups changes over time by comparing the theoretical frequency response curves with those that we observe. So some key features that jump out here are the existence of krill during winter, gelatinous species throughout the year, but especially in the summer. And we have fish throughout most of the record, but these are most abundant when the shelf edge current is weaker and when it's particularly strong, as during this period, which is why we have knockdown of the mooring, then the fish are absent altogether. And then I will say a few words also about the data from M1. So the ADCP data shown here demonstrate that the velocities are primarily oriented along the axes of the channel, such that we have for most of the year transport of water from the north into the Kvitoya Trough, but also importantly during the spring of each year there's a weak but persistent reversal in the current direction, and this influences the water column structure and also what we see in the water column. So here you can see temperature and salinity from the M1 mooring and the echogram, and all of these plots are overlain by the 34.7 isohaline. And then we have sea ice, day length, and water mass characterization below. And looking at the echogram in the middle plot, you can probably see immediately a lot of seasonality in both the density of organisms which are present, but also where they're located in the water column. And to summarize very quickly what we see, when there's an increased inflow of warm Atlantic water from the north, we tend to see more organisms in the water column. And when, on occasion, the Atlantic water or Atlantic influenced water disappears altogether, then we have sometimes a very sudden reduction in the amount of organisms which are present. But where they're distributed is also very variable. So during the winter months, there's a concentration of organisms within the upper polar water layer, whereas during the summer they're located primarily within the deeper warm polar water layer. And during the late spring and summer of both years, you see there's a seasonal minima in the concentration of organisms and very few organisms within the intermediate water mass. And you can see that more clearly here. Again, I've shown 5-day time slices of the echograms. Figure A shows a typical example during the winter months where you have an accumulation of macroplankton within the picnic line and fish schools in the overlying cooler polar water layer, and then a more diffuse assemblage of macroplankton in the underlying warm polar water layer. Figure B just shows the onset of the vertical migrations, which begin with the downward propagation of organisms located earlier in the winter within the picnic line. And in figure C, this is the typical situation during the spring or late spring and early summer when we have what appears to be an almost empty water column in the middle with distinct scattering layers near the bottom and near the surface and little evidence of interaction between them. And then when the ice disappears, we have a rapid increase in the density of organisms present. And in this example, you can see evidence of twilight migrations going on. So to conclude, the data that I've presented reveal considerable variability in not just the density, but also the vertical distribution of organisms throughout the season and over shorter time scales. And this is linked to physical conditions such as the changes on light, sea ice, and water column structure, but particularly the relative volume of polar water and warm polar water that's present at the site. And these data really highlight the tight relationship between the physical characteristics of the pelagic water column and the organisms that live there, and also demonstrate the complexity and organized structure of the pelagic biota. Thank you very much.
There is time for a question or two to Heather.
Hi, I was wondering, can you somehow relate your sound scattering layers in the two locations based on the kind of advected timescales? I mean, like temporal shifts or seasonal shifts, depending on where you are, where you see most of the zooplankton in the area, which can that be related to the kind of advective timescales of Atlantic water at those two locations?
Yeah, that's a very interesting question and something we haven't tried to do, but we should try to do. So yeah, thank you. I will look at that.
I'm going to be a bad moderator and steal a question here. I was just really surprised at the M1 situation where you had most of your organisms deeper in summer than in winter. That's kind of backwards to my reality.
Yeah, we are not seeing smaller organisms, so we're not seeing the plankton or even copepods or microplankton. So there probably are a lot of organisms up there, but why the larger organisms are deeper down, I don't know. I'm not a biologist, but...
Thank you very much, Heather. Any other questions from the floor?
I'm not really an expert on ADCP, but how do you distinguish if the signal or the scattering is coming from biological signals and not just particles?
So we're not using the ADCP to look at particles. There's a separate echo sounder, and then we can look at the differences in the if there's some confusion, we can look at the differences in the frequency response to identify, for example, bubbles from diving animals or microstructure. So yeah.
Okay, thank you.
I'd like to thank all the speakers, and I'm sure you want to join me in that. Mats and I are very, very pleased with how our Atlantification session has gone. And of course, that's a tribute to the speakers in yesterday and today's session for being so professional and rising to the challenge of addressing really short talks. You guys did a great job with our 10-minute time slots, and we thank you very much. I'd really appreciate it if today's speakers would come to the front of the room for a few minutes to engage with questions that we didn't manage to catch from the floor. Thank you very kindly.
No, too shy.
Interesting.